Welcome. In this session, I'm going to discuss about transcription. So, what is transcription? The process of copying genetic information from genetic information from one strand of DNA into RNA. So, is called transcription. So the process of copying genetic information from one strand of DNA into RNA is what I call transcription. Right? So here, in case of replication, we have seen the entire DNA is duplicated. But in transcription, so that will be not the case, right? So I just mentioned in case of replication, DNA replication, entire DNA is duplicated. But in case of transcription, only one segment of DNA is copied into RNA. So that is the difference between DNA replication and transcription. So in DNA replication, entire DNA is duplicated, but in case of transcription, only one segment of DNA is copied into RNA, right? So when we take, okay, so this is the DNA molecule, right? Say suppose from this point to this point, so there is a gene X. Okay, so after that there is gene Y. After that, no, there are no genes, and somewhere here there is gene Z and so on. Okay, right? Now say suppose only this part, that is gene X, is copied into RNA. Both transcription and translation are part of a protein synthesis, right? We know that protein is a sequence of particular amino acids. So a protein is a sequence of particular amino acids. So every protein will be having its own particular sequence of amino acids. Okay, right? So say suppose here there are amino acids like valine, okay, glutamic acid, then phenylalanine. Later I will give you all the 20 amino acids that are used in making a polypeptide chain. So say this is a polypeptide chain, and every polypeptide chain will have its particular sequence of amino acids. Then who knows to keep these amino acids in particular sequence? So right, so that is actually guided by mRNA. So that is messenger RNA. So the messenger RNA has information to keep these amino acids in a particular order, and then that results in the formation of a particular protein. Now, from where does this information on mRNA comes from? So that information on mRNA comes from DNA, right? So this is the DNA molecule, and from the DNA molecule you get this information. So gene X, here I said it is copied into RNA. That will be okay, mRNA. Say suppose, of course, later I'm going to discuss there are mainly three types of RNA that are required in formation of a protein. that we will see it later. Okay, so a particular gene, so only this segment, so from here to here is copied, okay, into RNA. And in some other cases, if this gene is required, that is gene Z is required, then this will be copied into mRNA. So depending on the requirement, okay, so particular genes will get copied into mRNA, and that mRNA will be translated. That results in adding of amino acids in particular sequence to form a particular polypeptide chain that becomes a particular part of protein, right? And while copying, okay, say suppose this gene X into RNA, only one of the strand is used to copy, right? So while copying, only one strand of DNA is used for transcription. Only one strand is used for transcription. So the question comes, what happens if both the strands of DNA are used for transcription? So here I'm going to take a, right, an example, right? So with some sequences. Okay, so these are the two strands, right? Now say suppose by using this strand there is a synthesis of mRNA, and now what will be the complementary here? So to this A, so it will be T here, but in case of RNAs there will be no thymine, there will be uracil present, okay, instead of thymine. So uracil is present instead of thymine. So thymine is seen only in case of DNA molecule, whereas instead of that thymine, uracil is seen mRNA. So hence I need to mention here U. For this G, it will be C, then G, A, then U. So this is the sequence on mRNA. Now say suppose this strand is also, right, transcribed, then what happens if that strand also transcribed? So now here this is another mRNA molecule, and this will have A, right, then G, C, okay, U, and A. Now you can see here these two mRNA molecules became complementary to each other. So these two mRNA molecules became complementary, okay, molecules. So they became complementary to each other. Now when we see these two mRNA molecules, when they become complementary to each other, so here the adenine will pair with the uracil, and you can see there will be, right, pairing, and this will become a double-stranded RNA molecule. So this results in the formation of double-stranded RNA molecule. When these two strands get transcribed, I told you that in case of transcription, only one strand of DNA is copied into RNA. But okay, if we say that both the strands get transcribed, then it results in the formation of two mRNA molecules which will be complementary to each other, and they will pair with and result in the formation of double-stranded RNA. And once the double-stranded RNA formation is seen, it cannot be used in the process of translation. So such double-stranded RNAs cannot be used in translation. So they cannot be used in translation. So there is, for translation to take place, compulsory the mRNA molecule should be present in single stranded form. It should not be present in double stranded form. So that is the reason why during the process of transcription only one of the strand will be used to make the RNA molecules. Now let us see in detail how this process of transcription takes place. So now we understood that during the transcription process only one strand will be used to make the RNA molecules. So now let's look at transcription unit. So this transcription unit, it has three regions in DNA. So there are three regions in DNA molecule, okay, right? And that will be a promoter region, then there will be the structural gene, and then there will be a region terminator region. So just now I told you, say suppose okay, so you see something like this, right? And I told from here to here is gene X. So this is gene X, then gene Y. Now this gene X will have a region that will be a promoter region. So every gene will have its own promoter region. So here there is a promoter region, and here will be a terminator region, and between these two will be the structural gene. So the structural gene is the one which actually codes for the polypeptide. So this gene actually codes for polypeptide, right? Whereas these promoter regions and terminator regions will help in identification for the enzymes. So there where will be the starting point of the gene, and where will be the ending point of the gene. So here in this case you can see this part, say this is promoter, and here is terminator, and between these two is the structural gene. So let's see the transcription unit. Right. So this is 5 dash to 3 dash, and this is 5 dash to 3 dash. Okay, so say suppose this is a region of DNA. So we are not studying the entire DNA molecule. I have taken only a part of DNA molecule, and in this DNA molecule, say suppose, so this is the region with some specific sequences, and it is called promoter region. So it's called promoter region. So promoter region will have some specific nitrogen based sequences, okay, that can be identified by the enzyme. So here there is a single enzyme, so that single enzyme used in transcription. There is a single enzyme that is used in transcription, and it's called DNA dependent DNA polymerase enzyme. So in the case of replication, in case of replication, we have seen enzyme called DNA dependent DNA polymerase, and in this case we see an enzyme called DNA dependent DNA polymerase enzyme. Simply you can call it DNA polymerase. In simple, you can call it DNA polymerase enzyme. And this is the enzyme that catalyzes the formation of RNA, okay, on one strand of DNA. So this is the enzyme that catalyzes the formation of RNA, right, on one strand of DNA. So enzyme that catalyzes the formation of RNA on one strand of DNA. So you can see this is a promoter region. Of course, whatever here I given you the regions, promoter, structural gene and terminator region, all those regions are just some particular sequences of nitrogen bases. Okay, so here is another region that's called Terminator region, and between these two regions is the structural gene. So from here to here is the structural gene. So that is the actual gene that will be coding for the polypeptide, whereas this promoter will be the starting and the terminator will be ending of this transcriptional, okay, unit, and this will actually help the DNA polymerase to identify the starting of the structural gene and ending of the structural gene. And here this DNA dependent DNA polymerase, okay, it catalyzes, okay, the formation of RNA. So it catalyzes the formation of RNA only in 5' to 3' direction. Okay, so earlier also when we seen in replication, DNA replication, that DNA dependent DNA polymerase also forms a new strand of DNA in this direction only, that is 5' to 3' means it is going to add new nucleotides on the third end, that will be the third OH end. Okay, so the formation of RNA will always take place from 5' to 3' because this 3' will provide OH ends, and these enzymes, whether it is DNA dependent DNA polymerase or DNA dependent RNA polymerase, will be able to add nucleotides only on the third OH end. Right now, out of these two strands, the strand on which the RNA is formed is called template strand. So it's called template strand, and this template strand will be having polarity of 3' to 5'. So it will have the polarity of 3' to 5' on which, okay, the RNA will be synthesized from 5' to 3'. The opposite strand is called the coding strand, and this will be having polarity of 5' to, okay, 3'. So out of this here, what I mentioned, so this one 3' to 5' is the template strand. So this one I'll just mention with the dot red colored area. So I'm just mentioned with that. So this indicates that this is the template strand here. Right, so this is template strand. Now opposite one will be the coding strand. So this is called the coding strand. So we have to keep in mind that the template strand will be having the polarity from 3' to 5', whereas the coding strand will be having 5 dash to 3 dash. So this is the coding strand and this is the template strand, and it is on the template strand, okay, the synthesis of RNA molecules will be taking place. And all the reference points while defining transcriptional unit, while defining a transcription unit, should be made only with the coding strand. So that's, I'll mention, all reference points while defining transcription unit should be should be made with the only coding strand. Means, so whenever you talk about this transcriptional unit, you should talk with okay, the coding strand as a reference point. Now if I ask a question that is the promoter present on the third end or fifth end, so the answer is the promoter is present on the 5 dash, because I told you to take the coding strand as the reference point here. Okay, so coding strand is the reference point, and right, the promoter is present on the 5 dash. So this 5 dash is usually called upstream, so it's called upstream, and this 3 dash is called downstream. So whatever I just mentioned here, so 5 dash is upstream and 3 dash is downstream. So with this we understand that promoter is present on, so promoter is upstream, whereas the terminator is downstream. So it is present on downstream, whereas promoter is present upstream. So wherever the 5 dash, this is fixed, the 5 dash is upstream and 3 dash is downstream. And while referring a transcriptional unit, you should be using the coding strand, right, as a reference point. That is what, okay. So we understood that the promoter is present on upstream and the terminator is present on downstream. Next, the process of this transcription involves 3 steps. So that will be initiation, then elongation, and then termination. So these are the 3 steps that we see in the process of transcription. While the transcriptional unit has 3 regions, that is promoter, structural gene, and the terminator region. So in the process of transcription, there must be initiation, elongation, and termination. And of course, all this is performed by, okay, the enzyme called DNA polymerase enzyme. RNA polymerase enzyme. So all it is performed by only one enzyme, DNA dependent RNA polymerase enzyme. Okay. Now this DNA dependent RNA polymerase, which I will be calling simply RNA polymerase. So when this RNA polymerase, okay, comes and binds to this promoter, okay, so this binding is aided by a specific protein called sigma factor. Okay, so sigma factor need to bind with this RNA polymerase. So this is RNA polymerase enzyme, and this is called the sigma factor. So this sigma factor, when binds with the RNA polymerase, okay, it changes its functionality so that it can identify the promoter region. So once the identification is completed, the sigma factor will unbind. So the sigma factor will unbind from the RNA polymerase, and now this RNA polymerase will be moving, okay, from 5 dash to, right, 3 dash. So because the template strand is, this is the template strand, and on the template strand the synthesis of okay, RNA molecule. So here I'm mentioning it mRNA. Not only mRNA, any other type of RNA formation is also called transcription, right? So here the RNA polymerase formed, right, mRNA in the direction 5 dash to 3 dash, and finally it comes to the terminator region. So when it comes to the terminator region, here another factor should bind, okay, with this RNA polymerase, that's called rho factor. It is called rho factor. So when rho factor binds to this RNA polymerase, it results in unbinding of RNA polymerase from the terminator, and thereby this mRNA will also get, okay, released from the template strand. And of course, one important point is that while this transcription is taking place, okay, I hope in earlier sessions I mentioned already, I'm repeating it again now, that the two strands should be separate. So here, while this process of transcription is going on, the template strand and the coding strand will get separated. So whatever that hydrogen bonds present between them, okay, so they get breakdown. So all will be break down, and you will see the template and coding strands will be separated, and then there will be synthesis of these RNA molecules. Okay, once this is finished, now the hydrogen bonds will form back. So in the beginning they all break down, and later once this transcription is completed, all those hydrogen bonds between the template strand and coding strand will form back, right? So whenever there is a need for this gene to get transcribed, the two strands need to get separated. Okay, so right, the DNA dependent RNA polymerase enzyme will catalyze this formation of RNA, and when it binds to this, okay, promoter, okay, there is a sigma factor that should bind to the RNA polymerase, so that the RNA polymerase can identify the promoter region properly. And once that is finished, the sigma factor will unbind from that point, and only the RNA polymerase will move over the structural gene, and when it comes to the terminator region, okay, the rho factor binds to it, and then with that, okay, the RNA polymerase will unbind from the template strand, thereby completing the process of transcription. And while, okay, elongation, so here when this RNA polymerase binds to this promoter, so that's what we call the initiation process. So that's called initiation. So in the initiation, right, the sigma factor binds to the RNA polymerase, and the RNA polymerase binds to the promoter region. So that's called initiation. And elongation, which means right here we see the RNA polymerase moving over the structural gene, and as a result what happens, the mRNA length goes on increasing. And of course, in the same way, in the process of replication I mentioned, the DNA polymerase enzyme adds nucleotide by nucleotide that results in increasing in the length of the strand. Here also the same process takes place. So there will be nucleoside triphosphates. So nucleoside triphosphates. So in the last session I mentioned what is nucleoside triphosphates, and they have dual function. So one, they will act as substrate, and also they are going to provide energy for polymerization, so in that, the last two bonds present between the phosphates are high energy bonds, and when they break up, so we know that, for example, adenosine triphosphate, right? So this adenosine triphosphate will break up to form, right, adenosine diphosphate, and you can see here the energy is released. Of course, in this case it is going to form adenosine monophosphate and 2pi, and this releases energy, and that will be used for, okay, polymerization process. In the same way, we'll be having, okay, CTP, right, and this will form into CMP plus 2pi, the same how we have seen in case of DNA replication. So here also we will be seeing the same nucleoside triphosphates, which will provide energy for polymerization, right? I hope it's clear. You can even take UTP, right, and GTP also in this case, right? And one more important point, so this DNA dependent RNA polymerase enzyme, so this is actually it is capable of, right, only elongation process, only elongation process. So it is capable of only elongation process, but due to addition of this sigma factor and then rho factor can alter its functionality, so that they can bind with the promoter, okay, and then even with the terminator, so they can identify the terminator region or the promoter region. So I have to keep in mind, the sigma factor is a transient, means it will be attached to RNA polymerase only for short period of time until the RNA polymerase binds to the promoter. Once it is finished, the sigma factor will unbind, and only the RNA polymerase will move on the structural gene, by, of course, during which there will be formation of mRNA, and once it comes to the terminator, then the rho factor binds, and finally the whole process, that is the termination. So in this case, the rho factor is required. For initiation, sigma factor is required. Next, only one RNA polymerase, only one type of RNA polymerase is present in prokaryotes. So only one type of RNA polymerase is present in case of prokaryotes. Okay, of course that I already mentioned. Now, but coming to eukaryotes, there is a division of labor, and we find, okay, three types of RNA polymerase. So three types of RNA polymerase are present in eukaryotes. So there are, okay, three types of RNA polymerases which are present in case of eukaryotes. In case of prokaryotes, only one. So that is which I mentioned here, DNA dependent RNA polymerase, okay, is the only enzyme that is capable of making mRNA, tRNA, rRNA and all. But in case of eukaryotes, that is not the case. So let's see, in this eukaryotes, those are called, as I mentioned, three types of RNA polymerase. One, it will be RNA polymerase 1, then RNA polymerase 2, and RNA polymerase 3. Okay, so these are three types of RNA polymerases in eukaryotes. Now let's see what they are going to transcribe. So this RNA polymerase 1, so it transcribes rRNAs. So it is going to transcribe rRNAs, ribosomal. So that small r indicates ribosomal RNAs. So like 28S rRNAs, 18S rRNAs, and 5.8S rRNAs. So those rRNAs are transcribed by the RNA polymerase 1. Now coming to RNA polymerase 2, it is going to transcribe, okay, HN RNA, going to transcribe HN RNA. So this HN RNA is a precursor of mRNA. So HN RNA is precursor of mRNA, right? So here HN stands for heterogeneous nuclear. So it stands for heterogeneous nuclear RNA molecule. So heterogeneous RNA nuclear. Okay. So RNA polymerase 2 transcribes okay, HN RNA. And coming to RNA polymerase 3, okay, it will be transcribing right, tRNAs. So here the T stands for transfer. So later I'm going to discuss about rRNAs and also tRNAs, and we will see in detail about these mRNA molecules, how they are present, right? So RNA polymerase 3, it transcribes tRNA, then okay, 5 sRNAs also it's going to transcribe, and then okay, SN RNAs also it's going to transcribe, right? So here the SN stands for small nuclear rRNAs, small nuclear rRNAs, 5S rRNAs and tRNAs are transcribed by these by this RNA polymerase 3. So 1, 2 and 3. Okay, that is seen only in case of eukaryotes, but in case of prokaryotes, only one type of RNA polymerase will be transcribing all of this, whatever, okay, whether it is rRNAs, mRNAs or tRNAs, all are transcribed by only, okay, one type of RNA polymerase. So that is in case of prokaryotes. Right, now let's see transcription unit and the gene. So the next one we'll see about transcription unit and the gene. We know that the gene is the functional unit of inheritance. So gene is functional unit of inheritance. So it's a functional unit of inheritance, and there is no doubt that the genes are located on DNA molecule, right? And it is difficult to define the DNA in terms of DNA sequences. I'm sorry, difficult to define a gene in terms of DNA sequences. Already I told you that a gene is a sequence of nitrogen bases. So whatever the gene, it will be a a particular sequence of each gene has its own sequences, right? So particular sequence of nitrogen bases. Now even the sequences, the DNA sequences that code for tRNA or mRNA, whatever rRNA molecules, they are also genes. Then okay, so here we will see what actually is a cistron. So this cistron is a segment of DNA that codes for a polypeptide. So it's a segment of DNA that codes for a polypeptide. So we know that I told you in a polypeptide chain there is a particular sequence of amino acids, and this information is present on mRNA molecule, and the information onto mRNA molecule, okay, is from the DNA molecule. Okay, so of course by transcription process the information from DNA is passed to mRNA, and through translation the information present on mRNA is used to make a specific polypeptide. So hence, okay, so when I take a DNA molecule, right, say suppose from this point to this point is a gene and this is a cistron. So it is a cistron, and that cistron codes for a polypeptide. Now, okay, the structural gene in, okay, in a transcription unit, it can be monocistronic or polycistronic. So structural gene in a transcription unit, so structural gene in a transcriptional unit, so this can be either monocistronic or polycistronic. So what we mean by monocistronic, so monocistronic, which may, of course, that I'll be discussing. Monocistronic is seen in case of right, eukaryotes. It is seen in eukaryotes, whereas polycistronic, okay, we will be seeing in prokaryotes. So monocistronic, okay, will be seen in eukaryotes and polycistronic in prokaryotes, right? And this part I'll be discussing in the next session. So I'll just mention, so monocistronic structural gene and polycistronic structural gene. Right, I'll be discussing about that monocistronic structural gene, polycistronic structural gene in detail in the next session.